Okay, so thank you for joining us for today's webinar, a masterclass on plants and nuclear waste, friends or foes, with Dr. John Pittman, and we're also joined by Dr. Andrew Lowe. So we're delighted to be joined by you both today. What I'm going to do is I'm going to very quickly run through today's agenda, and then we'll introduce ourselves, and then get started with the main content of today's session. So today we will follow up with a masterclass. There'll be plenty of time for questions and answers after that masterclass. And because this is related to our pollution and environmental control online course, what we're also going to do is give you a brief course overview and to give you more information about the programme. And if you do have any questions throughout, then I do encourage you to submit them via the Q&A function, which you can select in the banner on Zoom. And then we'll just follow up today with some closing remarks. So to get started, just to let everyone know, if we've not spoken before, I'm Hazel and I'm a course advisor here at the University of Manchester. And basically I'm here to help to give you the information that you need and to inspire you to hopefully choose to study with us. I work with our MSc Pollution and Environmental Control online course. And if you do have any questions, you can ask them during today's webinar, or you can also email me, or you can find out more information on our website too. And I'm available for individual chats if you want more information about the course that way. And all the contact details will be shared at the end of today's session. I'm going to now hand over to our panel to introduce themselves.
Okay, hello. My name is John Pittman. I'm a senior lecturer in the Department of Earth and Environmental Sciences, and I'm also one of the lecturers in the Pollution and Environmental Control Master's course.
Thanks, John. My name's Andrew Lowe. I'm the course director for Pollution and Environmental Control online, and I'm also a lecturer on the course here in the Earth and Environmental Science Department, specialising in hydrology and water resources.
Okay, so we're now going to hand over to John to start today's webinar. So John, over to you.
Okay, thank you very much, Hazel, and thank you everyone attending and watching in. So as I say, my name's John Pittman and I'm a lecturer with an interest in understanding how pollution impacts our environment, and in particular how pollution impacts biological components of our environment. And what I'm going to be talking about today is one aspect of that work, this interaction of the plants, a key component of our biosphere, with nuclear pollution. So before I start, I just want to acknowledge many of the people that were involved in this work with me. So the various researchers that performed the majority of the experiments, many of which did this as part of their PhDs. So Helena, Jeanette, James and Katie gained their PhDs through part of this work, and Philippa, who worked as a postdoctoral researcher. And indeed, Jeanette was also a previous student on the pollution and environmental control degree before she completed her PhD with me. And also listed here various collaborators, many of which also worked as co-supervisors for many of these researchers. So the interest here is understanding how radioactive pollutants interact with the biosphere, and particularly the interaction with plants. And the reason for this interest in considering plants is because plants play a central role as an entry point of pollutants into the wider biosphere environment. And this is because plants are primary producers of energy and nutrition. So all organisms, either directly or indirectly, utilize plants as a source of energy. And therefore, if that plant material contains pollutants, such as radioactive pollutants, there's then a risk that those pollutants could be transferred more widely into a food chain. And this is what we are referring to as this concept of food chain transfer. But the other aspect that is potentially of interest in understanding the plant interaction with radioactive pollutants is whether these plants could even be a tool to help us remove pollutants from the environment. And this is what we refer to as bioremediation. Radioactivity is an aspect that's critical for civil nuclear energy production. But of course, like a lot of activities, there's a risk that wastes are generated, either at mine sites, such as this site shown here in the top right, a site which I'll talk a bit more about in a moment, that is an abandoned uranium mine that we've worked on, but also at waste processing sites, such as this photo here of the Sellafield nuclear fuel reprocessing site in Cumbria. What do we mean by radioactive pollutants? So most of the elements in the periodic table can have radioactive forms. Radioactive elements or atoms have an unstable nucleus, and they therefore distribute energy, excess energy, in the forms of particles or rays. And we refer to these unstable elements as radionuclides. And they occur naturally, very commonly. So uranium is a very abundant element in the Earth's crust, and of course a critical component in nuclear energy production, which is mined and then processed. It's a challenging pollutant because of its duration. It has a very long half-life, and it will also then decay into other radioactive products that are commonly abundant in the environment, like thorium, radium, or radon, and others. But also we have radioactive materials that get produced during the reactions that take place within a nuclear reactor. And some of these can be very radiotoxic and very damaging, very challenging if they were exposed to the environment. So how could these materials become pollutants? Well, as I said, the initial starting point of the energy process is the initial extraction, the mining of uranium ore from the ground. And like any mining activities, this generates waste. So we have waste spoil, we have drainage waters. So there's always going to be a risk that some of this material will be mobilized into the wider environment. Also, the other downstream processes, so the milling, the refining, the processing of that material into components that are actually utilized in the energy production can also generate waste. And then we've got the reactor processes themselves and the waste that are produced during the nuclear reactions. Now, of course, at this later stage, these wastes are very highly regulated, and obviously because they're becoming increasingly more toxic, there's increasing control to prevent any of these wastes getting into the environment. So the major concern predominantly is waste that are generated through mining activities, either active mines or legacy mines. But of course, accidents, unfortunately, can happen. And we'll be aware of some of the more notorious accidents, so particularly the Chernobyl disaster that occurred in the mid-1980s in the former Soviet Union, this nuclear reactor that exploded. And slightly more recently, the Fukushima disaster in Japan that resulted from a tsunami affecting this nuclear energy site. And these accidents led to the release of radionuclides into the environment. In the case of Chernobyl, a very large area was contaminated, which gave rise to an exclusion zone that is still in place today, but also much of continental Europe was also contaminated by radioactive materials. Fortunately, in the case of Fukushima, the majority of the deposition was into the Pacific Ocean. So again, plants are at the forefront of mobilization of pollutants into the wider environment. This photograph here shows this famous area, the so-called red forest, just adjacent to the Chernobyl site, that experienced really high deposition of radioactive materials. And it was called the red forest because the trees changed to this red coloration due to the effects of the radioactive deposition. This killed a lot of trees, but actually a lot of plants materials did also survive, and there's been recently a lot of regeneration of plant material from this site. And this is one of the big challenges, because although these radioactive elements such as uranium, such as cesium, are not essential to plants, the plants can still nevertheless accumulate them, either through aerial deposition or particularly through accumulation from the soil via the roots and then transfer into above ground tissues. And this is because plants are unable to easily distinguish between essential elements that play a nutritional role and these elements that are non-essential. And if these plants survive the accumulation of these radioactive materials, this is when there's the challenge that if they are then consumed by a herbivore, such as an insect or an animal that is grazing on this plant material, there's this risk that they can accumulate this radioactive material. And this is what we refer to as food chain transfer. And there's been a lot of concern and investigation more recently with regard to this challenge in the wake of the Fukushima disaster, particularly with regard to radiocesium deposition. So radioactive cesium was released from the damaged reactor, and there was a concern that this could contaminate the agricultural system in the adjacent parts of Japan. So a risk of contamination to rice plants, and as well as grazing lands that animals would graze on and potentially give rise to contaminated meat and dairy products. Now, as it turned out, that the concern here was not as substantial as initially feared. While there was some levels of radiocesium accumulation into crop plants, it was much less substantial than feared, and was very much short-lived. And it turned out that actually over time, a lot of this cesium was getting locked into the soil and not being able to be efficiently translocated into the plants. But what about other elements such as uranium? I'll come back to that in a moment. But there's also this potential flip side, the potential positive of this accumulation of these pollutants into plants. Could we use this accumulation as a means, a natural means, to remove these pollutants from a contaminated soil or groundwater? And this is the concept of using plants for remediation that's often referred to as phytoremediation. And this is an aspect that's been investigated quite a lot over the years for a whole range of different pollutants, both inorganic and organic pollutants. And two components that are of interest in a radioactive pollutant scenario is a concept called phytoextraction. So this is the idea that the plant will be able to accumulate large amounts of pollutant into leaf biomass. Obviously, the plant has to be able to survive this pollutant burden, and then periodically the plants can be harvested, and then this material can be stored or dealt with, and therefore you're concentrating your pollutant in a single point source. The other concept is phytostabilization, where the pollutant can interact with the roots of the plants can be bound to these roots, and this can prevent the dispersal and the spread of a pollutant more widely in the soil or in the groundwater. So we've been investigating the interaction of plants with uranium here in the UK, making use of 2 field sites that are shown here. So these sites we're using as sort of natural laboratories to understand the interaction of plants with contaminated materials. So this first site is in Scotland. It's called Needle's Eye, so named because of this eye of a needle-like formation in the rock, which you may be able to make out in this photo. This is not a former mine site, but a site which, due to its geology, just has higher than normal concentrations of uranium due to the minerals that are present here. And then the second site, South Terrace, this was a former uranium mine site, abandoned about 100 years ago, and over time has revegetated, soil has been generated, and is a good site to understand interaction of regrowing plants with this contaminated land. Both sites have above background concentrations of radionuclides, including uranium, thorium, and radium. So we went to these sites, we surveyed the plant ecology at these sites, identified the most abundant understory vegetation. So at the South Terrace site, particularly these 2 plants, Asplenium and Primula, were abundant. At the Needle's Eye site, these 3 plants, Chrysosplenium, Iris, and Oenanthe, were surveyed. So we took plant samples, we brought them back to the laboratory, and we analyzed the uranium content in the plants and the different components of the plants. And what you can see from this graph is that the dark coloration indicates the presence of uranium in the root samples of these plants. And what we find is that predominantly the uranium is accumulating in root samples, preferentially in contrast to the aerial leaf and stem samples. And what this tells us is that actually this risk of transfer more widely into a food chain via consumption of leaf tissue is actually quite limited, because the bulk of this uranium is being locked into the roots. And we can also see this by looking at these autoradiography images. So these plants which are emitting energy due to radioactivity, if we place them on X-ray film, the X-ray film will be able to pick up this energy being released, and then we can visualize the accumulation of this radioactivity in specific parts of the plant. And you can actually see really clearly, particularly with the Primula example, the partition and the retention of this material, preferentially in the root samples. We can also look at this same data in a slightly different way, not by simply showing the relative proportion of the uranium in one part of a plant compared to the other, but by quantifying the ratio of the uranium in the soil versus the uranium getting into the root. And this is what we call a transfer factor. And what we can see in this graph is that the plants grown in the South Terrace site, shown by the S1 to S5, are taking up uranium into the roots much more than the plants grown at the Needle's Eye site. And there are other soil chemistry factors that differ between these 2 sites. So the pH of the soil is significantly different between the 2 sites, but also the organic carbon in the soil, the total organic carbon, is significantly different between the 2 sites. And indeed, what we see is that where there's higher total organic carbon in the soil, this reduces the amount of uranium that is able to get into the roots. So it's one factor that can help us understand what is the risk of transfer into plants, but also, of course, this can have a consequence for the use of plants for phytoremediation. The other soil factor that we've investigated is the presence of soil fungi, and a particular class of fungi called arbuscular mycorrhizal fungi. These fungi naturally associate with the roots of plants, and they form internal associations within the plant root cells, structures such as arbuscules and hyphae, and also externally, the hyphae of a fungi spread out into the soil, and they help the plant acquire essential nutrients such as phosphate. And we've evaluated the plants that we surveyed from our field sites and found that for all the plant species, they do indeed have the presence of these arbuscular mycorrhizal fungi. So they have the presence of the arbuscules, the vesicles, the intracellular hyphae, which are all hallmarks of the presence of this fungi. And this is actually fairly unsurprising because we know that about 90% of all land plants do indeed show associations with these class of fungi. But the question we've asked ourselves is, can these AM fungi play a role in controlling, potentially enhancing uranium accumulation into plants? So we tested this by setting up some controlled experiments where we took the soil from the South Terrace site, this soil that is naturally contaminated with uranium. We also sterilized this soil using irradiation to remove any microbial presence of this soil. So any fungi that are present in the soil was removed. We grow our plants in the soil, and then we can add back individual fungal strains that we've isolated from the field site and identified. And I'm showing you some of the data from these experiments here. What you can see in this graph is that indeed, when we add back individual AM fungal strains, we can enhance the accumulation of uranium getting into the plant. So the control is the plants being grown without any presence of AM fungi. And then we have 3 different strains that we've isolated that are naturally present from the field site. And we can see that they can enhance the accumulation of uranium into the plant. So particularly the Rhizophagus irregularis strain can substantially enhance the accumulation of uranium. So indeed, this is another factor that we know can control and can determine the ability of plants to accumulate these radioactive pollutants. And we've looked at this in a little bit more detail by using microscopy and imaging techniques to get a better understanding of where the uranium is being localized within these plant cells. And what these images here show that for the plants that have been associated with the AM fungi, the amount of uranium that we can detect in these root cells is higher. So what you have here is force color imaging, where the brighter colors, the kind of reds and the yellows, shows higher abundance of uranium in the outer surrounding of these cells. And we can even look in even finer detail using electron microscopy. And this is some quite new data. And what we are seeing here is that the uranium biomineralizes. It forms precipitates within the cell, specifically within fungal tissues of the cell. And these uranium particles are accumulating. We know they contain uranium because we can detect that by spectroscopy. And we can also show that these particles are also containing other elements like phosphorus and oxygen, so probably phosphate that's also present within these particulates, and also other elements like calcium. And what this suggests to us is that these biominerals that are accumulated in the roots are non-mobile, because they're precipitates, they're not in a liquid phase, and therefore this is likely the explanation for why there is reduced transfer of this uranium into the aerial parts of the plant due to this formation of these precipitates. So I shall make some concluding points to finish. So our research, and indeed research of others, have shown that uranium in many plants predominantly accumulates in the root tissues, and therefore this is reducing the risk of uranium being consumed by herbivores, eating that plant material, and reducing the risk of transfer into wider food chains. We've also shown that the increased abundance of organic carbon in the soil can also limit that transfer of uranium into plant tissues from the soil. And also our arbuscular mycorrhizal fungi, when they are associated with plants, these can also control the mobilization of uranium. It can actually increase plant uranium transfer, but particularly enhancing this transfer into the roots. And this seems to also give rise to a new form of biomineral aggregates that accumulate in these fungal hyphal tissues within the roots that we think are less mobile and explain the reduced accumulation of uranium into leaf tissues. And therefore, there may be potential of using these plants to concentrate and retain uranium in the roots to prevent their dispersal more widely in soil and in groundwater, and possibly for then recovery and storage. So I shall end it there, and thank you for your attention, and I'm more than happy to answer any questions that anyone might have.
Thank you very much, John. And just to say as well to everyone who's been listening, if you do have any questions, now is the perfect time to start submitting them. We've already had one question submitted, which is a question about what's the software used to create the visualizations throughout your presentation?
The visualization of the general presentation was just generated in PowerPoint. So it was PowerPoint that was used, if that was what the question means.
When it was submitted, it was around the time that you were showing the photos of the uranium accumulation in the plants, in the roots.
Okay, so the graphs themselves, I was using software called GraphPad Prism to generate those graphs.
Brilliant. Yeah, so we did have some clarity there related to the cells and the uranium throughout. So we've got a question of why does TOC impede uptake in plants?
So what we think happening is the uranium is probably binding to the organic carbon. So therefore, the uranium, we think, is predominantly being mobilized into the plant tissue when it's in a uranyl ionic form. And when it's bound to organic carbon and other soil organic materials, it's not then in a bioavailable form that is able to be transferred across the root cell membranes into a plant. And this is similar to what we find with a lot of mineral nutrients. They typically need to be in an ionic form that can be dissolved in the soil solution to allow them to allow these minerals to be transported into plant cells. So what we think is when there's higher concentration of organic carbon, this is reducing the amount of uranium that is in a bioavailable form.
Brilliant, thank you. And another question that we've had submitted, has there been any investigation done into how uranium affects the fungi directly?
There has been some work, not by us, but by other groups, and likewise the effect that uranium has on plants. So actually, we don't see any evidence of toxicity of the fungus to the uranium. This may be because the fungal strains that we've been working with are strains that we isolated from the mine site, and perhaps the fungal strains have adapted over time to the pollution conditions, the potentially toxic conditions of this site. And we do know that a lot of microorganisms can adapt to potentially toxic conditions, so that for the same species you can see variation in response. What we also know is that a lot of the toxic responses to elements like uranium are particularly due to oxidative stresses being produced. So rather than a radiotoxic effect, it's typically a chemotoxic effect that occurs. And it's typically the accumulation of reactive oxygen species that are formed within the cells that tend to give rise to toxicity. And I can hypothesize that these organisms that have adapted to these sites have adapted mechanisms to allow them to be more tolerant to these oxidative stresses.
Great. And this is the last one of our submitted. Oh, sorry, actually, we do have another two. So this is probably a good time to say, if you've got any more questions, please do submit them, because we've just got a couple of questions left. But the next question is, would you suspect that there's an adverse effect of uranium on the plant species diversity in the contaminated sites?
Yeah, that's a good question. So we do know that at some contaminated sites around the world, yes, this does have an effect on diversity, and those species that are more sensitive to these pollutants are not able to dominate. So again, it's a very difficult question to answer when you have a site that is a long been contaminated site. You can go to another site and compare, but it's not always an ideal control, because obviously the conditions can be different. So what we can do is get an overarching view of what would be the typical biodiversity that you would expect to see at one site compared to the other. Now, actually, at the field site in Cornwall we worked at, the biodiversity that we observe is actually not that unlike a typical understory vegetation. So what I mean by plants that are typically grown underneath larger trees. So we see typical plants like brambles and ivy, and these plants like primula. So actually the diversity was not too different. So I think rather than having a marked effect on the biodiversity, it's more that the plants that are there are potentially more tolerant to these conditions.
That's brilliant. So the next question we've had is, what's the impact of uranium in the human body?
Well, like any radioactive material, yes, it can. If it accumulates to high enough quantities, it can potentially cause cell damage that could give rise to the formation of cancers. It can give rise to tumors at a more local level. Very similar effects that we see in other organisms like plants and fungi. Again, these same oxidative reactions can take place that can give rise to damage. So damage to DNA that can cause mutations, damage to proteins, damage to cell membranes. So it's similar effects happening. But we also see similar effects with other metal pollutants, also metals like cadmium or lead or mercury, and they accumulate in animal tissues and human tissues. You can get similar effects, damage to cellular structures, damage to DNA. And it's this mutation burden that is also obviously the key challenge that can give rise to potentially to cancers.
That's great. And I think this is the final question that we've got submitted for now. There will be time for more questions if anyone thinks of anything after the next section of today's webinar. So if you do think of anything else that we've not answered, then please do still submit it and we'll come back to it at the end. It's just relating back to your the first question we had submitted related to the software. So saying that it was specifically referring to the data visualization tool used to create the picture of the uranium present in the cells, rather than the graphs. Could you give some more information about which software you used for that, please?
Okay, so yeah, so the uranium present in the cell. So the technique we used was a spectroscopy technique called nanosims. So that's single ion mass spectroscopy, and the machine that's used has its own bespoke software that can generate the images. I must admit I can't recall the exact name of the software, but it's software that's developed for the specific mass spectroscopy tool that was used here to generate these images.
Okay, that's great. Thank you very much. Yeah, as it was just a thank you as well. So thank you very much for clarifying all of that. So what we're going to do now is we're going to move on and provide a little bit more of a course overview about our part-time online Masters Pollution and Environmental Control. So what I'm going to do is I'm going to hand over to Andrew now to start to introduce the course.
Great, thanks Hazel. Thanks John, very interesting talk. So if you're interested in more interesting course units like John's, then we have this online course for pollution and environmental control online, and it's 100% online. We've designed it so it can be taken full-time or part-time. We've got students who are doing it in 2.5 years, 3.5 years, and you can go up to 5 years for that program. And we've designed it so it's flexible, so you can work at your own pace and your own time. So it's good if you're trying to combine a career and upskill yourself in a master's degree. So you can do it while you're working. And most of our students who are on the course currently, who are part-time, are also working. We do have full-time students as well. Next slide, please, Hazel. So just a selection of the course units you can take as part of this online course. John's talk today, he takes or is a course coordinator for the human impacts on the biosphere, and you've seen some of John's work that he teaches in today's webinar. Then we focus very much on measurement and prediction, so how we measure environmental data, how we're able to use that to predict environmental data. And then we focus very much on pollution, mobility and transformation, but also how we manage pollution. So my courses are on screen there. I teach 3 of those course units, specializing in the management of pollution, looking at the hydrosphere and using computer modeling to try and to explain environmental processes and how to remove pollutants from the environment. And we also look at air pollution as well. A real focus of the course is to undertake independent research projects. So we have environmental MSc tutorials that support those independent projects, and we do a range of different types of research. So looking at air pollution, looking at heavy metal contamination from mining, looking at the ecology and water quality. Also look at upland carbon flux, as well as looking at pollution in drinking water and soil contamination. So those are some of the flavors of research projects that we're doing on this course unit. Next slide. And here are some of the 2 current students and 1 past student with their comments about the online MSc program we run. They found it very useful. We try and design it so it gives you skills, practical skills in the environmental science field, focusing on pollution. And that's trying to give you skills not to progress onto a PhD if you're interested in research, but also if you want to go have a career in industry or in a government institution. So we try and design our modules to give you a good skill set as well as being interesting. So those are some of the comments there. Next slide, please, Hazel. So the entry requirements, well, we'd like you to have an upper second class honours degree in a subject that's related to the course, science-based subject. But if you don't have that, we do look at work experience. And if you've got a number of years of work experience, we'll also consider you based on your background. in terms of your work experience. We do have an English test requirement, so we need to score above 6.5 overall, and with no component below 6, score of 6. There's an email address that you can contact Hazel on screen there, and I think Hazel put that into the chat at the end of this this presentation. Next slide, please. And just on, I think this is my final slide, just on tuition fees, you can see the UK and overseas fees on screen. But if you apply by the 30th of June, then you receive a 10% discount on the fees. If you're a graduate of Manchester University, you will get an additional alumni discount, which is on screen. And this year, as of last year, we're offering a £5,000 discount to our international students to bring the fees down from 21,500 to 16,500. So that discount is in place for this coming September 2025. And then you can find out more details from the website, or contact Hazel on the email address that's on the screen. So I'll keep it short and sweet because it's been quite a long session, but any questions about the course, then I'm happy to take those questions. Thank you.
Yeah, and thank you very much, Andrew, for that, for running through the course information. And just to reiterate as well, if anyone does want to talk about sort of their individual circumstances, their course eligibility, you just want a bit more advice or to talk about the course in a little bit more detail, then you can contact me at studyonline@manchester.ac.uk as well. And if you do want to arrange a call, there is also the option for you to select her on the poll that I've just launched as well. So we've got one question that has come in about the course, Andrew, which is, in terms of the independent research project, what kind of options for hands-on research are available to online students?
Well, they're the same. This program runs on campus, so we have an on-campus version of this MSc program. So the online students have the option to undertake, at the moment, 2 focused study areas. So they focus at the moment on air pollution, so looking at machine learning and data science with air quality modelling in Manchester. And then there's the option working with myself and Dr. Steve Bolt, looking at upland carbon flux, where we're looking at the use of modelling and measurement to investigate the amount of greenhouse gas fluxes that are released from peat catchments in the Peak District near Manchester as they degrade. So there's that option. And we're trying to bring online the other areas of research, such as looking at ecology and water quality, looking at mine contamination, heavy metals, and looking at the drinking water, the water quality of drinking water and soil water quality. Now, those are currently this year are not open to the online students, so we've got 2 options for the online students, but we do plan to bring online the other projects very soon. But under those topics, you're able to specialize your independent research projects. Does that help?
Yeah, no, that's brilliant. Thank you. And thank you for taking the time to kind of give the more detailed answer there as well. And if you think of something after today's webinar as well, then you can always get in contact with us and ask those questions. We're happy to answer anything as you that might pop into your head in sort of half an hour, this evening, something like that. So if you do have any further questions, there's always a chance to get in contact with us, and we do welcome you to do so. So please don't be shy about that in the slightest. So I think that probably brings us to the end of today's session. So what I wanted to do is thank both John and Andrew for taking the time today to speak to us, and to you, our audience, and for those great questions as well. It's really great to hear you so engaged in John's research, and also to have the questions regarding the course too.